that I'm really excited that we finally got the presentation working. And I'm excited to be here, and I thank Anne and Melanie for inviting me. So I want to tell you a little bit about myself, and Anne did a great job. So I was an undergraduate in microbiology, and I had never heard of food science. So I heard about food science when I was a senior. Fall of my senior year, I took an applied microbiology class. And they talked about industrial applications in microbiology. And when they got to the food part, I just got excited. And they actually talked about one of the most boring things in food microbiology, and that's what got me excited. And when I used to teach it, I used to dread that part. It's like so boring. But it was in fact how we can foods and how we determine the heat transfer so that Clostridium botulinum spores are killed. So we open a can of foods and we think of it as this inert thing that's always safe. But it in fact has a lot of science behind it, and that's what makes it safe to eat. That's what got me excited about food science, so I started looking at programs with strong food microbiology, and University of Minnesota was one of those programs, which was convenient because I'm from Minnesota. So I went there for my master's, and for my master's I worked with an organism called Staph aureus. You hear about staph infections. Staph can also cause foodborne infections or foodborne intoxications, so that staph grows in food and can produce toxins. If you consume the food with the toxin, it causes 2 to 6 hours vomiting and diarrhea, the 2-bucket disease coming out at the same time. How I know this for sure is I contaminated myself with toxin during my master's, and it was kind of interesting. It's like, okay, this is going to happen next. And, oh, I got that symptom. I wouldn't recommend it to anyone, but that was my master's. For my PhD, while I was doing my master's, I met another faculty member in the department, and I took his class, and I loved it so much, and I asked lots of questions, and he invited me to join his lab. So for my PhD, I worked on fermentations, and I worked on cheese starter cultures. And it turns out that the bacteria that make cheese have certain, we call them phenotypes, certain characteristics that are needed for to make cheese and make it well, and they're unstable. So that my PhD advisor, Larry McKay, was able to figure out why the characteristics were unstable. And it turns out in that type of bacteria, they have things called plasmids, and a lot of bacteria have plasmids. E. coli can have a plasmid or two. Lactic oxide, the name of the organism that we use for cheddar cheese fermentation, have like 7 or 8 plasmids within the cell. And depending on how you culture it, they can lose a plasmid. So they can lose the ability to utilize lactose, so they can't ferment and make it anymore. Or they can lose the ability to break down the protein, so that they can no longer grow fast enough to produce good quality cheese. So that's the sort of work I did for my PhD. I've been at UMass Amherst now for 28 years. And in my time here, I have been doing research in the area of foodborne pathogens and antimicrobial delivery systems. So how do we add an antimicrobial into a food product so it's stable and inhibits microbial spoilage of pathogens? And my current work, I'm working on non-water-based cleaning and sanitation, which sounds crazy because you need water to clean. However, there are some food products like peanut butter or chocolate where they don't want any water in the manufacturing environment. And so my lab is working on formulating things made out of food-grade oils so that we can do cleaning and sanitizing in those processing environments and do it more frequently. So that's just a little bit about myself and my research, but I was asked to give you an overview of food science. So I can go down deep and I can do an overview. I don't have a whole lot of slides. So if you have questions, feel free to ask them as I go through it. Because an overview, we're not going to go very deep. But I do want to talk about food science. So food itself is something that we all see. And we usually see it in the grocery store, right? We go into the grocery store, but there's a whole supply chain. It starts with seed supplies, and that could affect food science. Maybe you're going to make frozen strawberries. And there may be a variety of those strawberries that hold up better to the harvest and freezing, right? So that would influence the final quality of the product. Or a variety of tomatoes that's better for canning, right? So there are different varieties that will affect the quality of the food as it goes through the entire time. Food scientists, we're really more focused on the food from the cultivation through the consumer and then what it does in the body. And the reason we're interested in cultivation, I told you I do food safety work. Think about who's harvesting the food. What are the food safety on farm practices? And we have an extension faculty member, Amanda Kinchla, who really focuses on this in our department. We're also concerned about processing. How do we process the food? How do we make it safe? How do we preserve the nutrients? How it's distributed, is it refrigerated distribution? What's happening in the warehouse? What's happening in the retail space and the consumer. If I had my notes, I'd give you some razzle-dazzle about how much the food supply influences the gross national product. And it's something like close to 10% of the gross national product is dependent on food. So people talk about food science, well, it's such a small science, but it really isn't. It's a big science. And it's an interdisciplinary science. So we study food from the farm to the fork. And food scientists, if I walk around my department, we all have an area that's kind of what we consider our specialty. So we're chemists, microbiologists, engineers. We have extension specialists, nutritional biochemists. And Alyssa will talk later. She's a sensory scientist. And we work together to make our food safe, tasty, and healthy. If you go and look at the food industry, they would divide the food scientists up by where their job is. So there are food scientists who work in product development. They're working on formulation of the food product. There are people who are working in manufacturing and the processing plants. And then they also have quality or safety specialists. There are also, we think about food in the grocery store, looking at all these brands. You see that, but there's also the food industry that's supplying all the ingredients that go into each of those food products, right? So it's just not the finished product. It's everything that's going in as it's coming out. So you'll find food scientists working in academia, food regulatory agencies, and food is regulated by the Food and Drug Administration. All processed food is regulated by them. Meat and meat products are regulated by the USDA. And so you'll find regulatory agencies both at a national level and state level and all throughout the food industry. So that when you go into a grocery store and you see your foods, they all have some part of food science associated with it. Now some of you may be looking at this and like, Lynn, I'm not into processed foods. Not cool at all. Well, processed foods are all foods, including things like an apple or lettuce that you purchase, okay? Food science itself integrates principles from biology, microbiology, biochemistry, molecular biology, chemistry, toxicology, engineering, sensory science, nutrition, and I probably left out a few, So we're very interdisciplinary, so we take basic science, and what holds food science together is the application of food. So if you put a bunch of food scientists in the room, and we were looking at this apple, each food scientist may say something different. The food chemist might be interested in the different sugars that are there. They might be interested in that acid. There's malic acid in an apple. They may be interested in the fibers. So there's insoluble fiber, cellulose. There's also soluble fiber, pectin. They might be interested in taking that pectin and using it as an ingredient in another food. You might take yogurt and one of the ingredients is pectin as a thickening agent coming from the apple. So the food chemist is interested in that. The food chemist is also interested in the three-dimensional architecture of the food. Why is it crunchy? Is there turgor pressure? Is there water that's keeping those cells crisp? Right? A food process engineer might think about how is that harvested? What are we going to do with it once it's harvested? We're going to wash it. We're going to take field heat off of it. Maybe we need to coat it with a little bit of wax so that it has a longer shelf life. When we're buying apples in June and they say New York apple, they weren't harvested in June, folks. They were harvested back in October, right? How are we going to store them that long? It's actually refrigerated storage with modified atmosphere. So they take out the oxygen and the CO2, and they also filter out something called ethylene, which softens fruits and vegetables. So even though you're looking at this, and this is not a processed food, we have lots of science going on, right? Then the sensory scientist might say, well, what does the consumer like? Do they like this type of apple more than another? Is it about the taste? Is it about the texture? It's about the color. When my kids were little, they wouldn't eat any apple that if you cut it, had more like a yellow flesh, because they associated it with, we got a Macintosh tree, that they hated the soft texture of the apple. So these are things that are important with the apple. So I'm a food safety specialist, and you look at an apple and say, what can be wrong with an apple? It turns out there are lots of things that happen with the apple. That when they're harvested, they're washed before they're distributed. And when you wash any fruit and vegetable, you want to make sure that the water that you're washing it in is 10 degrees colder than the vegetable. You need to take out the field heat. So the food safety specialist works with the process engineer to take out the field heat. And the reason why you want to take out that field heat is that if it's a tomato or an apple, if it's warmer than the water, it actually sucks the water in. So if that water's contaminated, maybe one apple out of 100 is contaminated, and it goes into that wash water, those organisms can come off. And if it's sucked in, you just contaminated the whole batch, right? And then the other thing we do is we make sure there's some sort of antimicrobial. So if anything comes off the apples, it's not spread to all the others. So I look at the apple and I think of potential food safety issues. There was an outbreak probably 4 years ago associated with caramel apples. And they think that it was both field heat, then the heat of the caramel warmed it up so the bacteria could grow better. And also, if it's not clean when you put the stick in, you're just shoving those bacteria into the interior part of the apple. So there are lots of things to think about when you look at something like an apple. And last, I have a health and wellness scientist. So in our department, we don't have, we have people who are biochemists who are really interested in the effect of the nutrients and the other ingredients in the apple on the body. So they might be looking at how does fiber affect the microbiome, or how the coloring agents, the flavonoids, affect the health of people who are eating the apple. So there's one apple, and it's like the blind people who feel different parts of the elephant. We all see different aspects of that food. In our department, we've divided, I don't have my notes to let me know where I'm going, so I'm just a little blind here. I'll tell you a little bit about our department. So UMass Amherst, we are the oldest food science program in the United States. So that we're the first established food science program. And currently, as of 2022, we're the top ranked program in the US. And we're ranked number 3 globally. So that's exciting. So we're at number 3 globally. The next ranked program in the United States is Cornell Food Science, and I think they're ranked 15 globally. So hopefully we're going to stay number 1 in the US for a while, and we'll see where this goes. So within our department, and I don't remember if it was Anne or Melanie asked me to address questions. And so one of the questions are, what are the big questions this field is asking? And I think a lot of the big questions are covered by what we think of our academic strength. So we take our faculty and we're kind of saying, okay, you're more interested in the molecular properties of the food. That's a lot of the food chemists would fall into that area. I think we have Alyssa in that area, but she would argue that she falls under foods for health and wellness more. So, and reality is, all of us collaborate with all the others. And so that's why this Venn diagram, I think, is a pretty accurate way of thinking about our food science program. Microbiologists fall under the safety of foods, but we also have a chemist who works on detection of contaminants. And you could argue she falls under the safety of foods, right? She looks at finding pesticides and nanoparticles in produce. Foods for health and wellness, I mentioned we got biochemists, nutritional biochemists who work in this area. And we have people who work what we're calling sustainable food processing. They're interested in some of the up and coming food processing things. And I'll talk about those more in a second. So you notice here, you can't see this, give me a second. Right in the center, this is what I call innovation. And this is really where we do our interdisciplinary work. The majority of the faculty at UMass Food Science, most of our grants have another faculty member on it. You have the PI, you have the co-PI. But the strength is that we don't forge forward by ourselves. We do it as a team, and we get that interdisciplinary research. One of the reasons we're number 1 in the US are based upon these 3 faculty members. So Heng Zhao, he works on diet and health. I would call him a nutritional biochemist. Eric Decker, he's one of the world's foremost lipid chemists. So he studies fat and how fat spoils and becomes rancid and how to prevent that. And then Dr. Lynne McLandsborough is an emulsion chemist who does so many different things. But if you ask him what he does, he says he does food nanotechnology. That's because emulsion drops are really small, so he can say that easily. But what he does is he's an amazing, almost physical chemist who can figure out how to formulate and structure foods. And the way our research works is I want to highlight one of the papers that they published last year. It's something like this. So this is a paper that was published in a food science journal, Food Chemistry. And the title is Black Pepper and Vegetable Oil-Based Emulsion Synergistically Enhance Carotenoid Bioavailability of Raw Vegetables in Human. So basically what they did is they designed a human study. And they asked people to eat salads, with and without a vegetable oil-based emulsion with black pepper. Salad dressing. But we have the oil chemist, right? Because oil and vinegar is salad dressing. Black pepper is a food product they're adding. And basically, they're then feeding people a salad that has tomatoes and carrots. I don't know if it had radishes, but picture kind of worked well enough. And then after they ate it, they took blood and they looked at carotenoid levels, vitamin A levels within the blood. And what they found is when you had the vegetable oil and the black pepper together, that you enhance that bioavailability of those nutrients. So a lot of the people who do nutrient work in our department think about how foods can be formulated to enhance absorption, right? So this is kind of an interesting thing. Ever since I learned about this, I pepper my salad generously and not feel too guilty about throwing on that salad dressing, right? So I'm making those nutrients more bioavailable. So that's kind of how the department tends to work, is we have this collaborations, we make each other stronger together. The other big question, any questions about that before I move on? Yeah.
What does the black pepper have to do with it?
To be honest, do you know?
Yeah. It is activates the trigeminal system, so it is like activates the TRPV1 receptor, and it's believed to help your blood flow. So it is located in your, I guess they might have their own theory, but based on my knowledge, it can be help with blood flow and with the absorption of the beneficial, I guess, vitamin A in this case, during not just in the mouth when consuming, which is when you actually feel the oral burn, but those receptors are in your GI tract, in your gut system, so it's thought that they're involved in that uptake. Yeah. So you could do the same thing with capsaicin.
Right. Right. Yeah.
That would be
Yes, it is spicy.
So, any other questions? Yes?
If you know, what made them think of it?
They had other studies before this where, and I was trying to find it because they had some really cool pictures, and I didn't know what the title of the study was and I couldn't find it, where they looked at different oils and how it affects absorption. And I think they did it in an animal model. And they were able to show that as you absorb fats, you get these, your bile salts that are secreted into your intestinal system actually makes an emulsion. And that they were able to show that that emulsion was absorbed differently depending on the type of oil. And it turned out olive oil enhanced absorption. So they've been working in a similar area together, the three of them as kind of a team. So that they've, found interesting things. A lot of what Dr. McLandsborough does is looks at how you can formulate these emulsions and can you actually deliver things directly to the intestine or where it goes after. So he is an emulsion scientist, but he's not just making salad dressing. He's making some very sophisticated emulsions where they'll add different proteins that will drop off at different pHs and really deliver it in the body. So, that's good. Any other questions? Ask me now. Yes?
What is field heat?
Oh, field heat.
Field heat. Field heat is the heat of the vegetable after you harvest it. So like when you pick a tomato in the hot summer, that would be the heat on the produce. And there's a lot of emphasis. It's probably 10 or 15 years ago. Do you remember when there was a big outbreak of listeria from cantaloupe out of Colorado? One of the things the company did is they didn't take the field heat out. So when they harvested, the cantaloupe were warm. And then they didn't add antimicrobials into their wash so that they think that it was sucking the bacteria into the surface. Yes?
I'm just curious, what kind of antimicrobials that would be food safe are you guys talking about?
They're adding, you can add bleach-based, so hypochlorite can be added at a legal level. And it's a low level. It's 20 to 200 parts per million, which is much lower than what you sanitize your bathroom with. Okay, so it's a very low level. It doesn't necessarily kill the bacteria on the surface, but what it does is if bacteria float off the surface, it kills them in the water. They also use something that's even better than that, which is called periacetic acid, or proxyacetic acid. And it's a combination of acetic acid and peroxide. And that is more effective. The problem with bleach is it gets neutralized if there's a lot of soil in the water, but it's not very stable. So that peroxyacetic acid is something that's used pretty universally in produce right now. Okay, future directions in food science. Yes.
Was the self-contamination during your master's intentional?
No,
I thought it was too.
That was the project. That was a thing that happened.
It was, so. No, it was accidental, and I was handling a concentrated solution. And I think what may have happened is maybe I generated an aerosol that I inhaled and then swallowed. That's, I was being very, very careful. But, and I never told my advice. I'm pretty sure that I've had graduate students get sick in my lab and they don't.
Right.
Yeah. Or they tell me 10 years after they graduated. So some future directions that food science is going. We're moving towards some green technologies and sustainabilities. I'll talk a little bit more about that in more detail. Food and personalized health and wellness. Personalized health is an area that NIH is really emphasizing. And so food will be part of that, right? Because food is so much a part of our health. We're moving towards integrating data and using smart technology in the food industry. And there is a move to modernize some food safety. I'll talk a little bit about each of these things. So for green technology and sustainability, part of this is agricultural practices. You know, how can you till the land so that you minimize the release of carbon dioxide. Organic farming is part of this movement, minimizing use of pesticides, water use during manufacturing, right? So I just talked about washing apples and washing equipment. Food processing is a water-intense environment. And so I think in the future, they're gonna start looking at, can we reuse the water? Can we treat the water, reuse, use it longer? And then also one area that my department is fairly active in is alternative proteins. And I'm gonna focus a little bit more on alternative proteins. So alternative proteins are basically looking for non-animal protein sources. So agriculture, our carbon footprint, the major carbon footprint is from animal production, mainly cattle production. And so there's looking at ways that we can look for protein sources that will not be as strict on the environment. So people think about plant-based proteins, and I have this image where there's Brazil nuts, those are low water use nuts, some garbanzos, soybean, lentils, sunflower seeds. So these are all things that could be used for protein. But reality is, my daughter is a weightlifter, she's a powerlifter, and she consumes protein like crazy. And it doesn't look like this. She has powder. So there has to be some technology to extract the protein out of the food. That's food science. Then once you get that protein, there has to be some technology How do you make it into a burger? How do you get the texture? Once you do that, do people like it? That's where Alyssa comes in, right? So there's all sorts of technology in all these different alternative proteins. What makes me laugh about alternative proteins is the difference between a food scientist and a nutritionist is the nutritionist will, I've heard nutritionists say this, eat no food product with more than 3 ingredients, and anything that's processed is bad. And I look at everything's processed, even the apple. And alternative proteins are gonna be the most processed foods you physically can think of, right? Because you have to go from that to a powder, right? So it's gonna be ground up, it's gonna be extracted, it's, fiber is gonna be taken out of it, sugars are gonna be taken out of it, they're gonna precipitate that protein, it will still be wet, they're gonna spray dry it to make it a powder. And now you have your powdered protein. It's not a panacea. It will be processed, and there's lots of opportunity for food scientists. So these proteins can come from plants, they could come from fungi, they could be extracted from mushrooms. Microbial fermentations is an area that we have a faculty member who's working on organisms that utilize helium and produce, and can live on helium. Makes me a little nervous to have him up there in his lab with his helium generator and growing the organisms. Trust me, environmental health and safety's been all over us. We have triple, quadruple fail safes, and hopefully all will go well. Cultured meats are something that we're hearing more about, and insect proteins. And one thing food science students do is they participate in product development competitions. So, the baking industry has one, Ocean Spray has one, and then the Institute of Food Technologists, who is our scientific organization, they also have a number of these product development competitions. So the students form teams, they work in our pilot plant, we have a pilot plant in our building, and they come up with food formulations, and then they compete against other universities. So a couple years ago, we had a team that was making a food product with insect. It had to use cricket powder. And they developed, and cricket powder is challenging because to me it tastes like dirt. And they ended up making kind of like a tomato paste in a tube, but they made it based on sweet potato, and it was supposed to be a product aimed for people in Africa to increase their nutritional protein content. And so I think it was sweet potato based, and then they added the insect protein to that, and it had kind of an umami, something to add to your stew to enhance the flavor. It wasn't a bad idea. They were one of the finalists, they didn't win. But every time I tasted it, they'd be like, here, taste this. So there it is. There's the challenge to the food scientist. How do you take an insect powder? What could you do? Maybe the food chemist would ask that. What do I need to take out to make it taste good? So the food chemist would take it out, and then they'd bring it over to Alyssa, who would run a sensory panel, and people would report, does it taste like dirt? Or not? Or how much do you like it? Would be probably what they would answer, yes.
Do you have to say yes every time one of your students wants you to taste something?
No, I don't have to. We have informal tasting, and then Alyssa will talk about some of the formal tasting that's done in the department. And there have been a few studies that she's done, and I've been rejected because my taste buds are too old. Which I just find kind of insulting. Yes.
So one thing is that vegetarianism is a great excuse to get out of some of these things, just saying. The question I had though is, so number one, there's the plant-based, and bottom one, insect proteins. Is there like a major advantage to processing these things into something else? rather than, I mean, people eat tasty crickets all over the world, like crunchy snacks, and,
Right.
you know, all those things are edible as they are.
Right, but a tasty cricket is still processed in some way, right? So it's been dried. I've had them, I was in a meeting and had to eat one to get a stuffed toy for my daughter when she was little. And it had nacho cheese on it. So I think that as we move forward with these technologies, the most important thing, people aren't going to eat anything that doesn't taste good, right? It has to taste good. Ideally, it should be nutritious. And it has to be safe. And to be honest, I don't know how much work has been done on the microbial food safety of just a dried cricket, because drying something doesn't kill the bacterial load. So that, as I said, you put food scientists in a room, everyone will have a different opinion, and that's my opinion on that cricket I ate. Yes.
Do you guys do any partnering with? I mean, obviously you're working with people in agriculture, but given like the current needs of the earth around climate change and global warming, and I'm just thinking about like almond milk, which I have every day, which I'm like, oh, this is terrible for the environment, right? So looking at sort of these high protein, you know, different foods, but thinking about them being centered like regionally and globally. So is that like work that you guys are taking on or thinking about?
It's something we discuss. We don't have a lot of expertise in our department in kind of supply chain of the food industry. And I don't think right now anyone in the, so resource economics does a lot of that sort of research. But it's something we do toss about, like, could this be a future hire? Is there some way we could do supply chain and integrate it into the department? Yes.
So you had mentioned, so like in this protein sort of variety, that Brazil nuts are like a low water option. But is there any sort of trade-off with can't you give yourself selenium poisoning with Brazil nuts?
I think that is a possibility. In addition, they're all grown in tropical areas and would have to be shipped. All right, so there is a trade-off. And that's where it's really hard if you dive into the research. If you're not, and it's really the economists, because anytime they calculate costs, so it's economists working with people who do more environmental, trying to figure out what is better for the environment, because it's really hard to figure out where they've taken the assumptions. So they have lots of things they assume when they do a lot of those calculations. And I don't have the expertise to, I've tried to really evaluate those. Alyssa, would you answer that differently?
No, I do have a great review research article on that topic that just addresses the concern for like differences in transportation, but the big effect has to do with water usage and greenhouse gas emissions. So while it still might have transportation, it's still really beneficial in those other areas.
Yes. Does the Harold's flavor competition make up for the cricket?
I'm lactose intolerant, so I sit there with a pile of lactates and try to taste their ice creams. I try to be polite. I do want to talk about cultured meat. I don't know if any of you read the New York Times this morning. Right?
Yep.
So here it is. U.S. approves sale of lab-grown chicken. So a couple weeks ago, they came up with the name, and I think it is laboratory grown meat. So it has to be labeled. It's like cheese food. Cheese food is cheese. It's made from cheese, but they've added additional ingredients, so it's now called cheese food. And there's actually a standard of identity. A lot of foods, mayonnaise, peanut butter, they have legal standard of identity. Ice cream, and ice cream has premium ice cream, standard ice cream, right? Low fat ice cream.
Yeah.
So there are legal standards of identity. So they just approved the sale of lab-grown chicken. And this is basically tissue culture. So they're taking the tissue from chicken and they're then growing it in culture to create the meat. I'd be curious, I'd try it. So they're gonna sell it at 2 restaurants. One of them is gonna be in Washington DC. Maybe next time I'm in DC, I'll try to find it and give it a whirl. But
Let us
this
know
is
how much it costs.
yeah. So this is the thing. I did tissue culture when I did my postdoc, and tissue culture, everything has to be sterile. So to sterilize something, you either have to filter it, and usually for a large fermenter, it's filtered. But those are oftentimes disposable. Or you have to autoclave it, right? So then you're using high heat. You have to use a modified atmosphere to grow the tissue culture. To get any 3D structure, there has to be a lot of agitation going on. So that there's a huge energy cost in order to grow it. And then the next question is, what are they growing it in? What's the media? Usually has to have a lot of protein, and it may not be plant-based, right? So it's kind of I'm not sure how the logistics of this will work. I'd be curious though, and I'd love to try it. I'm not going to, so the Institute of Food Technologists has an expo every year and scientific meeting, and I won't be going this year, but that would be a place you probably could taste it, yes?
This is probably a weird question, but like, I'm just thinking of lab culture, you're gonna put it probably in a petri dish. Do you guys also worry about the leaching of the plastic, like into like the food, or into the?
I would guess to make something at a large enough scale that you could produce food, that they're gonna be using stainless steel vessels. They would have to figure out a way to get away from the disposable plastics. But then your stainless steel vessel, how are you going to sterilize it, right? And a lot of this technology is available through the pharmaceutical industry. So our insulin we use is in fact a fermented product. They took human insulin, cloned it into E. coli. They grow up the E. coli and extract out the insulin. But it's easier to grow bacteria than it is food-based meat. So I thought that was interesting and timely that I saw this morning. When we think about alternative proteins, regardless of the source, there are a number of challenges. We need to look at the proteins, identify them, characterize them. And part of that is, if I'm going to manufacture something from food and I have an ingredient, that ingredient should be consistent, right? So I need to have markers so that each time, anyone ever make chocolate chip cookies and they turn out slightly different every time you make them? It's kind of like that. Welcome to the food industry. But when you buy an Oreo, it's always an Oreo, right? Or a Chips Ahoy. So it's about how do you formulate that food product, how do you deal with it as you're growing it so it's consistent. And part of that is understanding what's in your proteins. How you formulate it and process it. You want to think about production costs and supply chain. That would be the lab-grown chicken. It's, how much is that going to cost to produce, and can they produce enough in order to make it feasible? There are food safety and nutritional aspects. There have been a few outbreaks associated with cashew cheese, where manufacturers weren't paying attention to food safety and then got in problems. So we keep an eye on that. And the most important thing is consumer acceptability. So if you can make lab-grown chicken, but it doesn't taste like chicken, it could be that people who don't eat chicken because they're against killing any animal won't have any problem with it. It still tastes good, but it doesn't taste like chicken, right? But if you want to save the world and if it's more sustainable, you want to convince someone who loves chicken to eat this, because it's better for the world, it has to taste like chicken. Right? So it's the consumer acceptability really drives all of this. And it's a circle, right? First you have to see if it's feasible, you have to understand what's happening scientifically, and it may not be in this order, but it's trying, all these things contribute to whether or not it will be a feasible technology and if it will move forward. In the area of personalized health and wellness, there's been a lot of focus on the microbiome. And the microbiome are the communities of bacteria that live within our intestines. Most of the focus has been on large intestine, but people are also starting to do some work with small intestine. And the work they've done has shown that the populations of bacteria in our intestines may vary from person to person, but functionality, we have lots of bacteria that break down fiber. Well, those are more common in people who eat a lot of fiber who might be vegetarians, right? So the populations, it's not, Joe and Dave, but Joe and Dave both break down fiber, and they're more likely to find those two bacteria in people who eat a lot of fiber, who are nutritionists. So the diet shifts the population. So the bacteria in our intestines, something like 10 to the 12th bacteria per gram of poop. So that's a lot of bacteria. And most species are just bacteria by the time it comes out of our body. So we're giant fermenters. And when you shift what you eat and the population shifts, it doesn't necessarily mean that some bacteria go away. It means they have lower populations, but they're still there. And so that as you shift your diet, maybe go back to meat or high fat, they may grow higher, and the ones that break down fiber will go down. So it's a fluid population dynamics, and it really depends on what they can break down and how you deliver it. And so that's where prebiotics are. And prebiotics are basically looking at functional food ingredients that then modify your microbiome. So this could be fiber, fructooligosaccharides will enhance growth of things like bifidobacter. So we have a faculty member in our department who works on the microbiome of infants and how human breast milk influences that. So, and what he's found is it's a lot of the very specialized sugars and nitrogen sources that will change that population of bacteria. Other work we do in our lab or in our department are we understand absorption and functionality of food components. So that's a lot of Dr. McLandsborough's work along with Dr. Zhao. They do functional design of food products. So that's thinking about how you can design maybe delivery of a nutrient so that you enhance absorption. And here's Alyssa's work, treating loss of taste and smell. And you'll cover some of that a little bit?
I think I was asked to talk more about sustainability in plants. I can certainly talk about that. If there's time.
So this is one area that Alyssa works at. So when COVID hit, they reached out, and the group of people around the globe who work in this area got together and did a number of surveys and helped really figure out that, in fact, loss of taste and smell was associated with post-COVID infection. The other thing that's happening is there's more integration of data and smart technology in the food industry. I think what we'll start to see is integration of smart equipment or AI for food processing, where during the processing they'll monitor moisture or color and they can adjust the temperature of ovens. So this is an interesting field that's starting. Nimble manufacturing, this is something that's come out of COVID. Do you remember during COVID when they were dumping milk? And they were dumping milk because a lot of the milk was meant for institutions, so for school lunches. And for school lunches, or even at UMass dining, the milk comes in the big tubs, and you put it in the dispenser, and you pour it out. But what happened was they didn't have enough capacity to switch the milk and put it into cartons. And milk also has a finite shelf life, right? It will spoil. And so they basically were dumping milk because the equipment, the manufacturing equipment, couldn't handle the shift in focus. So thinking about manufacturing, oftentimes manufacturers, they set something up, to make one size package. But can you make machinery that could be more nimble? Can we shift more quickly? And then there's a lot of talk about blockchain for food distribution, and that's similar to AI technology, but that is really looking at, what are the numbers of packages being manufactured versus in distributor as it goes out to grocery stores, so that there aren't bare shelves, right? Keeping that distribution going and making sure that the data from the manufacturer and the distributor. So this image here, this is a grocery warehouse, and this is how groceries are stored before they go to Stop and Shop or Big Y or wherever you buy your groceries. And years ago, I took a field trip of students to one of these warehouses, and they had us sign our life away that you could be in danger. And what they do is, let's say Stop and Shop needs tomato sauce, Kraft macaroni and cheese, Campbell's soup, this, that, the other thing. They have people on forklifts, and they are paid by how many pallets they can wrap during their shift. So they race around these warehouses, and they're picking things off of top shelves, lower shelves, and they're packing them onto their pallets, and then they're loading them onto trucks. And the students and I would be they'd come by, we'd be up against the wall, petrified. It was one of the scariest, I never went back, one of the scariest field trips ever, but I think it was probably one of the most informative to really see that in between. And I think there are a lot of people who work in the food industry who don't take into account the distribution of food. And I've heard horror stories of food companies who changed to a more sustainable package. And then they realize they don't fit in their boxes, so they get new boxes. And then they realize they don't fit the standard pallet to the same extent, and they just blew their entire supply chain when they should have been asking that question when they looked at their packaging. So there's a lot of things that go into product development. And everyone's heard that same story, and so now everyone's like, oh, it's right for the supply chain. So that's what this is. You don't think about this as food. We're also thinking about modernizing food safety, and a lot of this has been pushed by the FDA. So there's technology now where you can do whole genome sequencing of a bacterial cell, and you can basically get that data very quickly. And so what the FDA is starting to do is when they inspect food companies, if you have a food that they would consider potentially risky, if it's a food that's been associated with outbreaks before, if it's a refrigerated food, if it's a frozen food and you don't cook it ahead of time, they may go ahead and do something called a swabathon. So they start looking for pathogenic bacteria in the environment. No outbreak, we're just inspecting. And the problem for the food industry when they're doing that is you can't sell that food, right? Because you don't know, they might find something. You can't sell that. So it's a food industry is not happy about it. But the FDA is coming in, they're doing the swabathons, And if they say they find Listeria monocytogenes in a floor drain, they let the place know. The place will then be like checking everywhere for that strain of Listeria. They're gonna clean it up, they're gonna check their product, make sure it doesn't have it before it's released. Or maybe they won't release it until they've gotten rid of it in the environment. And then they start manufacturing again. 3 or 4 years later, if someone gets sick with Listeria monocytogenes, now when you're sick, the hospital has to provide the strain to the state public health, who then does whole genome sequencing and puts it in the database. And if there's a match between the patient and that Listeria that was isolated at this factory 3 years ago, the FDA will be in that factory looking for it. So in the old days, you would get sick. Listeria monocytogenes, usually people are immunocompromised who get it, pregnant women, and it can take a long time. It can be a short incubation period, but up to 2, I've seen even 3 weeks after consuming the food that you become sick with it. So what would happen is, I'm sick with Listeria, I'm in the hospital, because most people are hospitalized. When you get sick, it's not diarrhea, it's more systemic meningitis, septicemia. It's serious infection. And so public health will come up and say, okay, what have you had to eat in the last 4 weeks? Have you had any greens? Oh, were they bad? Oh, bad greens. Really? What brand? Where'd you buy it? And they'll keep probing. They might even show up and start swabbing your refrigerator looking for it. But you don't necessarily always remember. So the data is very wonky. So if we can have, it's kind of like a library of sequences that the FDA's found during inspections when a patient gets sick, at least I have someplace to start, right? Someplace to start looking. And it's supposed to modernize and make things happen faster. They've been doing it now for about 4 years, maybe a little faster, but not always. So it's still yet to be seen. And there's still outbreaks. Salmonella is the one that's really hard to type. And there are still outbreaks of salmonella where they never figure out the source. The other thing that people are starting to look at is non-enrichment based pathogen detection. And we have a faculty member in our department, Matt Moore, who's a virologist. And you can't enrich a virus. So an enrichment is, let's see. So I have an image there of a Petri dish. I can't see it, but I look at that, I can smell it too. But that's what they look like when we grow them in the lab. And like each one of those colonies, to get that big mucoid glob of bacterial cells, they're probably 10 to the 12, 10 to the 14 bacteria right there. The problem with food, and when you get an infection and go to the hospital, you have an infection, so that bad bacteria, the pathogenic bacteria, is overgrowing. So it's easier to detect it, right? If you have diarrhea, you sample the diarrhea, and that pathogen that shouldn't be there is there in really high numbers, and they find it. But in foods, we're looking for human pathogens that are in the food, but are there in really potentially low numbers. So there have been outbreaks of salmonella where there are less than 1 salmonella per gram of food. So the difference between talking about high numbers of bacteria, 10 to the 9, 10 to the 14, versus 1 in a gram is a huge difference. And so when we go looking for organisms in foods, bacteria, we take 25 grams, or if we're doing meat, we'll take a 65 gram sample of that meat, we'll add it to broth, we'll grow it up overnight, and let that pathogen grow to really high numbers so we can detect it. And that's been the benchmark. Matt Moore, who works with viruses you can't enrich. So he's working on technologies, nanopore sequencing, that can go after and detect things that are there in really low numbers. So it's trying to get past that gap. And we've been trying to get past non-pathogenic-based pathogen detection for 20 years. But I think working with viruses that can't be enriched is really gonna push it forward, and hopefully we can get it working with bacteria too. So a little bit how food science research is conducted. I just said that basically we do all the science related to food. So we do wet lab. There's a lot of instrumental analysis. Our department, and each faculty has their own thing. Like Dr. McClemens, the emulsion chemist, he likes equipment. He loves equipment. You go into this lab, there's like no bench space other than equipment. So we have tons of instrumental analysis available. We have faculty who do a lot of molecular biology, DNA sequencing, and then we have big data based on that, right? So then we have to do the data analysis. We have faculty who do simulation and modeling. So we have Dr. Jaikai Lu. He's a mechanical engineer and teaches our food engineering in our class, or in our department. He takes a lot of data and then models it. So, for example, we have a grant together where we're delivering nanobubbles of hydrogen peroxide for cleaning produce. And, excuse me, not hydrogen peroxide, ozone. So we're basically using ozone nanobubbles. And we're designing the experiment, so we're doing wet lab, but we're designing it in a way that we can use the data to model it and predict whether or not a treatment would work, and then we'll confirm that. We have faculty who do animal work, so a lot of the nutritional biochemists will work in mouse models before going to human feeding studies. Our human subjects can be feeding, it could be sensory, it can also be just surveys, right? Looking at base food safety knowledge. And then we also have people who do some database analysis, and Alyssa will be talking a little bit about that. One of the questions I was asked was, what kind of instruments are used? So this is a word cloud. I went on our webpage where we listed all our equipment, threw it in to get. I took the words out, instruments, systems, and there was one other. But this is pretty much what you see. We have a lot of HPLCs. The food chemists in our department use HPLC to separate proteins or lipids and characterize them. We have a lot of microscopes. Some are light. Dr. He does Raman scanning Raman scanning microscopy. So there are lots of different types of microscopes. We have an electron microscope, lots of incubators. Many things are automated, but this is basically what we're seeing. So we have lots of different equipment in the department. Nowadays, a lot of things are done in microtiter plates, so it's miniaturized, so there's less waste of expensive reagents, but there's also plastic associated. So to bring it back to the library, I wanted to talk a little bit about, I asked my faculty, how do you use the library? And this is the list I got. So we use journals and textbooks pretty much extensively, very extensively. Subject areas, there are some journals that are really food science journals, and there are some that are truly chemistry journals. And then there's like food chemistry, right? Food microbiology journal. So there's a variety of different journals that we use. A lot of us use the interlibrary loan of books and journal articles that UMass doesn't have a subscription to. And we use classroom textbook holds so that students have access to textbooks. We also use a lot of the university databases and software. Web of Science was something that a lot of faculty use. Your AOAC methods, so we have a subscription to that, and we use that for our analysis class. And you use it in class in lab, so that students have access to that, and we also use it in research. The Intel databases, which are more marketing, but there's also a lot of data there, and Alyssa will talk a little bit about that. And some of our faculty use the citation management systems. Personally, I've been using EndNote now for 30 years, and I can't wean myself off it. But some of the faculty in the department do use the library. I also got some other things. UMass SOAR, which is supporting open access research, which is a place you can apply to get funding towards publication of open access. Some of our faculty have used that. And a few people listed the UMass Writing Center, which is not, it's in the library, but it is not funded by the library. But that is another service. I looked it up. That we use. To dig into this a little more and have an example of kind of where we publish, I looked at our microbiologists. So I looked at 4 food microbiologists. Dr. Sela, he works on the microbiome work. Dr. Gibbons works on food fermentation and looks at the evolutionary development of things like fungi that are in Asian food fermentations. So things like soy sauce or use these fungi. So that's his area of research. Matt Moore, who's the virologist, and myself. So I looked at publications from 2021 till current. And these were the different publishers of the journals that we published in. I then did a further dive and tried to figure out how many different journals and which publisher we're using the most. I thought this might be interesting for librarians. The reason why I took the food microbiologists is of all our department, we publish and we publish well for microbiologists, but it's not like the chemists. If I did this for the chemists, it would have taken me 2 days, right? So our chemists, I think Dr. McLennan published 150 articles last year. That's just one of those. He's the most cited agricultural scientist in the world. And if I had done 3 years of data from him, I would have been going bonkers. So I did the microbiologist. It would be interesting. I was kind of curious. I'm like, oh, this would be interesting, but it's not that interesting. So out of the 4 of us, we had 1 publication in PLOS and 1 with a Wiley Black Belt journal. We had 2 journals that were published by Taylor and Francis. 3 from ACS, American Chemical Society. Remember, these are microbiologists publishing. And some of those were collaborations with our chemists, right? 4 publications each with the ASN journals and with Springer Nature. 5 through MDPI. 9 through Elsevier. And 11 publications through Frontier. And this surprised me.
Yeah.
I would guess if you looked at our chemists, they're going to be very heavy on Elsevier. Because if you publish 150 publications, you're not going to want to pay publication fees, right? And ACS journals. So this is just an example of a subpopulation of our department. So we're publishing lots of different places. And if you look at the journal titles, they don't necessarily say food science, right? So that these are also the journals we're using for research, reading the research of our peers that we're basing our projects on, because science isn't done alone. You build on what other people have done. There were also some publications in society journals. So, Journal from the American Society of Nutrition, American Academy of Pediatrics, and so on. So we did have some more society journal ones. And this is where I'm going to end my talk. I hope I didn't go too long. But there you have it. Yep. We have time for questions.
2 minutes for questions, but remember, she'll be here later after Alyssa's talk, so we'll have more opportunities. But if someone's got a burning question right now, we'll take it.
Yes?
What's an HPLC?
It's a high pressure liquid chromatography. And so you can take a liquid solution and put it through a column. And as it goes through the column, the smaller things go faster and the slower things lag, so that you can separate it just over a distance. And it will vary based on what's your carrying liquid and so on. So that we have both preparative HPLC, where you separate things out into different sizes, and then we have it attached to a mass spec, where it will separate it, and then it will break it up, and it will detect the different molecules. So you could detect a carboxyl group or a methyl group, different organic groups, so that you can try to identify what that molecule is. So there's a lot of high technology. And an instrument like that, an HPLC with a mass spec on it, is like a $500,000 instrument. It's not a small thing.
Sometimes that instrument keeps working perfectly, but it can only talk to a computer that's running Windows 95.
Yeah, that's very true.
Okay, so we are at time.